Next speaker for the afternoon is Dr. Bettina Lickiss. She's a very experienced scientist with a keen focus on the technology of induced pluripotent stem cell-derived cardiomyocytes. She's business development manager at InVitro, which is a contract research organization with a primary focus on delivering cardiac risk evaluation for new drugs for a preclinical safety, toxicity, and drug discovery. So without further ado, Tina, thank you very much for joining us today, and we look forward to your talk.
Thank you very much, Will. Thank you very much for the kind introduction. And also, you've basically taken all the very important information away from me already. So I'm a business development manager from InVitro, and we are a CRO service company providing cell-based assay services. And this is why my talk title is today Overcoming Cardiac risk-related obstacles with using cell monitoring technologies, because this is exactly what we're doing. We're providing cell-based assay technologies, and I thought today we have quite a few little projects I would like to show you. And the idea is, unfortunately, it's not moving, unfortunately. So maybe I can just talk a little bit more while this is getting sorted out. So the idea was basically that we have a lot of clients that ask us, oh, that was quick, that ask us a lot of questions, of course, when they approach us. And the top 4 questions are basically how the next projects basically resulted in, why we performed the projects, because of course we want to give the right answers for that. So one of the very typical questions, of course, if you have a cell-based assay system using human iPSC-derived cardiomyocytes a lot, is how relevant is the data that you're producing? Is it comparable to animal models, etc.? And this is something we have addressed nicely with, of course, going back to the CEPA study analysis with typical compounds that you know from the CEPA study that were used to address this issue, but also outside of the CEPA scope. So we'll show you a little bit about that. Second question we get a lot when it comes to people working with with chamber-specific diseases like atrial fibrillation or ventricular-related diseases, then they would like to know, how can we address these? Are the commercial iPSC-derived cardiomyocytes around? So that's what we also did, and we performed a study on contractility using chamber-specific cardiomyocytes. The next question is also very typical, is when we use commercial iPSC-derived cardiomyocytes and then we basically run out of a lot, it's how do we have to worry about the lot-to-lot variance or not? And this is what we did then with another project where we addressed 10 different cell lots from the same vendor and checked the contractility on pre- and post-compound analysis. And last but not least, is it possible to analyze compounds with different MOAs simultaneously? Which is basically meaning how fast can you be actually? How quickly can you analyze our compounds? And this is something we've done with the AtlaZ technology, and I would dive into that at the end. So because the first 3 questions are dealing with the FLEXcyte technology, I would like to give you a little introduction about the technology itself. So the key parameter you can see here are the membranes of the FLEXcyte plate. They're of typical human cardiac physiology. So the cells are plated on top. They can contract, and when they contract, they lift the membranes upwards, and therefore you can see the electrodes in the video there in the device. The change in distance is then measured via capacitive distance sensing. So we have a physiological membrane, and we can monitor. So it's not an endpoint assay. We can monitor the functional toxicity the whole time, and of course, because it's contractility we're focusing on, force is the main parameter. But besides force, we of course also look at all the other important parameters, which is beat rate irregularities, early after contractions, for example, which would be like the downstream effect of an early after depolarization. We have downstroke slopes, upstroke slopes, durations, of course, area under curves, all being separated into upstroke and downstroke level. So a good idea for you how this technology works. And then we can dive in straight into the first question that I said, like this is basically the question that get asked all the time. So we had a closer look at cardiac risk-related compounds. We started with 2 typical CEPA-related compounds. One is of high risk, one is of low risk. So sotalol is high risk with a potential for duration prolongation. Nifedipine, not really. And I think you can see here very nicely on the left-hand side. This is basically a spider plot because I wanted to prevent you having like thousands of slides to just show you the different parameters. Therefore, you just get a good glimpse at every parameter. 100% is control, basically. And you can see for the duration that we have an increase on a concentration-dependent level for sotalol. But for nifedipine, we have rather the opposite. We have a decrease on a concentration duration dependent level for the duration. So the focus of the CEPA study. Moving out of CEPA and into just different compounds, in this case antibiotics, we've chosen ciprofloxacin and moxifloxacin. Wanted to show that we have a same class drug, but working a little bit differently on an MOA level. Ciprofloxacin is known to be not so harsh on the cardiac risk-related level, whereas moxifloxacin is a bit stronger. And this is what you can see very nicely here on these spider plots as well, that we have concentration dependence. Ciprofloxacin data is pretty much the same. But with moxifloxacin, we do get some special data on the beat rate variation and the durations and area under curves as well. So diving straight next into the next question, the CHEM-specific diseases. How can we address these with commercial iPSC-derived cardiomyocytes? Because usually they're of mixed cell populations, so they usually provide HL cells, ventricular cells, nodal cells. But here we assessed a commercial cell type that was just ventricular, just HL cells, and compared these with the FLEXcyte. We started with a pre-compound analysis, meaning that we looked at the general beat rate, and you can see here that the atrial cells performing much higher than the ventricular cells. So there's an indeed difference between the cell types. And if you look at the contraction shape, like the mean beat, as we like to say it, then you see that we have a very typical shoulder for the ventricular cells that we do not see for the atrials. So this was the pre-compound analysis, and then we started with carbachol, which is a acetylcholine receptor agonist, which works predominantly atrial. So we wanted to see the effect on the atrial cells that we did. We have a concentration-dependent increase in the duration when we apply carbachol to the atrials. If you look at the ventricular cells, you do not see such a strong effect on these cell types. Well, seeing that the duration increases, you would expect the beat rate to go down, of course, because it just needs more time until the next beat can happen. And you can see that here at the beat rate over time. If you look at the carbachol data, this is basically the analyzed data, then you see that we have a duration or a negative chronotropic effect of carbachol on the atrial cells and not such a direct effect on the ventricular cardiomyocytes in comparison. Another compound that we looked at is assay, and in this case, we decided to go with the raw data because you can see actually very nicely what's happening after 1 minute and after 10 minutes. So starting with the control condition in the first panel, you can see once we apply the compound, we have an increase in the duration directly for both cell types. But after 10 minutes, we have a very, very physiological effect on the ventricular cells, where we see that the duration actually decreases again and gives rise to a higher beat rate. What we do not see with the atrial cells when you compare the beats basically in the window with the control situation. So that was just a glimpse of what we have performed with the CHEM-specific cardiomyocytes. Coming to the next question, the commercial human iPSC-derived cardiomyocytes, how reliable are they? This is a very typical question when people have one lot and then they ran out of it and then they get a bit worried thinking, okay, now we have to go to a different one. Does that going to ruin my data? So what we did was an analysis of 10 different cardiomyocyte lots of one vendor from FCDI, and we did a functional performance in this case. So we have a pre and post compound analysis in this case as well. We always do the same procedure, seed the cells 5 or 6 days in culture, and then we can perform the analysis. We have nifedipine and sotalol here as the gold standard compounds that we used. And the pre-compound beat rate here shows you very nicely that we have a very consistent level of all 10 lots. They were, by the way, not produced in 1 month. They were produced over a time frame of 2 years, so very consistent. And these beat rates did not vary much. So it was between 47 and 53 beats per minute. So this is basically how we started. And then we added nifedipine to the cells. And you can see here, different colors represent different concentration ranges, with highest concentration in pink. And we see here very nicely that we have a concentration-dependent decrease of amplitude. The upper and lower bars here should actually be dashed lines showing the standard deviation, because the standard deviation is very low, but the presentation mode kind of kills that. And So we're very happy with this. And what you cannot see with this kind of setup is the effect on the duration. That's why we have a little snippet here to show you that nifedipine has indeed a duration decrease upon higher concentrations. So next compound in this case was sotalol, where you also see that we have actually no effect on the, no strong effect on the amplitude at least, a very good standard deviation below 10% as well, but of course an effect on the duration, where we see that we have here with higher concentrations of sotalol, a contraction duration increase. Right. So I'm running quite through because I know you're tired and I don't want to keep you for too long. So last question, is it possible to analyze the compounds with different MOAs? And in this case, we introduced the AtlaZ system because we know we can do a comprehensive analysis of it. And to give you a little bit, again, I mean, it's been mentioned before today, but because it's rather electrophysiological topics here, I go into the details of how it works again. So the nice bit about it, and regarding the question of how fast can we be, is usually we work with 96 wells, but in this case, we have the possibility to basically analyze 6 times 96 wells simultaneously. So we get, of course, quite a bit faster. The technology is impedance-based, so we have the typical setup that the gold electrodes apply a current, and when the cells go on top, and they basically form a monolayer and impede the current, you have a rise in cell index. And once you apply something, a compound or a CAR T cell, etc., that affects this morphological changes, then you see a decrease in the cell signal. And so the parameter again is monitoring. There's no endpoint to it. It's just label-free monitoring of what happens to the cell on a morphological level, let's say. And you can measure, and that's the great part about the AtlaZ, the frequency level from high to low or from low to high. So basically starting with little things like barrier integrity, going to higher frequencies like higher than 40 kilohertz, for example, and then really look at cytolysis, etc., like membrane changes, etc. And what is also great about it, which is actually, I think, like the key thing for saving time on a CRO perspective, is that you do not have to choose one of the frequencies to go forward. You just analyze everything at the same time, and in the end, have a look at your data. So our study design in this case was, because we had 6 times 96 wells, we did not choose human iPSC-derived cardiomyocytes. We just chose a cell line, H9C2, cells, and put basically differentiated them in-house towards cardiac phenotype by serum starvation. Then we added the compounds, and we chose a selection of the hazy cardiotox study because it's a nice selection of compounds with different mechanisms of actions. So you get a good overall view of drugs that would affect mitochondrial level, drugs that would affect on I don't know, on morphological level, etc. And then we tested that for 3 days and put it in the monitoring mode in this case to analyze the effects of all these agents. Now, I'm not going to shock you with 8 of them. I'm just make a selection starting with pentamidine because I think it's interesting, because actually in this case, it should not be a control situation, but in this case it is, because H9C2 cells do not have any hERGs, so there can't be any blocks, so there shouldn't be any result, and there is no effect on it. If you see it on the cell signal level on the left-hand side, the dashed line shows when compound addition is applied, and you see that there's no decrease in cell signal upon addition of pentamidine at any kind of concentration, basically. Another kind of a positive or a control situation is erlotinib. It's a cardio-safe compound that should not have any cardiotoxic effect on the cells, and this is also what you can see here after compound administration at any kind of concentration. It does not harm the cells on that level. But if you go into, if you apply vincristine, a very cardiotoxic agent, to the cells, you do see that we have a strong concentration-dependent decrease in the cell signal on the left-hand side once we apply the compound, and also on the analyzed level on the right-hand side. You can see very nicely the significance towards the control situation. And last but not least, we of course applied also doxorubicin, a very typical anthracycline, very cardiotoxic, and see a similar effect that we've seen with vincristine, a very strong concentration-dependent decrease in the cell signal upon application of this compound, and see on an analyzed level as well how this looks on an in comparison to the control situation. And with this, I would like to thank you very much for listening at that late time of the day. And thanks, Nanyon, of course, for being our great partner and for inviting us, and all our collaborators where we had the great data done with Fujifilm, Cellular Dynamics, Axel, Nexel, Cellogics. Thank you.
Thank you very much, Bettina. That was really nice and awesome to see some new AtlaZ data as well from our new instrument there. Really nice to see that on screen. Now, you and I talked beforehand. In the interest of time, we thought that we would forego questions for this talk. And then if anyone has any further questions, please feel free to contact Bettina after the session.
All right. Thank you very much.
Great. Thank you. One more round of applause for Bettina. Thank you.